Good morning. I'm Dr. Lavanya. I'm an assistant professor in Department of Biochemistry, Mallareddy Institute of Medical Sciences. Today, we are going to discuss about the vitamin B6 that is pyridoxine. So, vitamin B6 is compound. It has three compounds: pyridoxine, and pyridoxamine. So, when it comes to chemistry, they are the pyridine derivative. Pyridoxine is a primary alcohol, and pyridoxal is an aldehyde form. Pyridoxamine is an amine. So, pyridoxamine is mostly present in plants, while pyridoxamine are also for presently animal foods. Synthesis of the coenzyme. The active form of vitamin B6 is the coenzyme pyridoxal phosphate. So, pyridoxal phosphate can be synthesized from three compounds like pyridoxine, pyridoxal, and pyridoxamine. So, vitamin B6 um, is excreted in urine as 4-pyridoxal acid. This is the structure of pyridoxine, pyridoxal, and pyridoxamine. Here, the pyridoxine, in presence of enzyme pyridoxine kinase, gets converted into pyridoxal 5-phosphate. So kinases are useful in attaching the phosphate group. And phosphatase removes the phosphate group and converts pyridoxine phosphate to back again pyridoxine. So this pyridoxine phosphate in presence of vitamin B2 uh, gets converted into pyridoxine phosphate prime 5 phosphate. So then pyridoxal is a aldehyde form. It's also by pyridoxine kinase gets converted into the pyridoxine phosphate, and phosphatase is helpful in converting back the pyridoxine 5 phosphate to pyridoxal again. Pyridoxamine acted upon by pyridoxine kinase and is converted into pyridoxamine 5-phosphate. And the phosphatase can useful in converting the pyridoxamine phosphate back to pyridoxine. Absorption. The mainly pyridoxine absorbs in the proximal jejunum by passive diffusion. So, in the mucosal cells, all the forms of pyridoxine are converted into pyridoxal so that it can be easily absorbed. The pyridoxal form can be easily absorbed from the proximal jejunum by passive diffusion. Transport. The pyridoxal is transported in circulation by binding to the albumin. And storage. Mainly pyridoxal phosphate is stored in tissues in the form of its coenzyme. It is mainly stored in liver, brain, kidney, and muscles. Excretion of 4-pyridoxic acid is excreted in urine as such. The functions of vitamin B6. There are many as a coenzyme, it mainly acts in the various metabolic reactions, mainly involved in the transamination, deamination, decarboxylation, and condensation reactions. So functions, the pyridoxal phosphate is a metabolically active form of vitamin B6, and liver is a site of vitamin B6 metabolism. So the primary role of vitamin B6 is to act as a coenzyme in coenzyme to many other enzymes in the body that are involved in predominantly in metabolism. The metabolism of what is a protein, carbohydrate, and fat synthesis and breakdown. The synthesis of a neurotransmitters like serotonin and everything are important for the nerve function. The synthesis of histamine also involved in allergic immune response. Hemoglobin synthesis and its function, hemoglobin carries the oxygen to tissues and carbon dioxide to the lungs. Gene expression, which is involved in the protein synthesis in the body. So biochemical functions. Pyridoxal phosphate is closely associated with the metabolism of amino acids. And synthesis of certain specialized products like serotonin, histamine, niacin coenzymes from the amino acids is dependent on pyridoxine. This histamine, serotonin are the new. Serotonin is actually a neurotransmitter. It is helpful for the transmission of the impulses. Pyridoxine phosphate participates in various other reactions like transamination, decarboxylation, deamination, transsulfuration, condensation, etc. Transamination, the pyridoxal phosphate converts amino acids to the keto acids. The keto acids enter the citric acid cycle and gets oxidized to generate energy. The vitamin B6 is an energy releasing vitamin. So during transamination, the pyroxyl phosphate interacts with amino acid to form shift base. The amino group of is again handed over to pyroxyl phosphate to form pyroxamine phosphate and keep the keto acid is then liberated. See, L-glutamate and pyruvate in the presence of alanine transaminase or the glutamine pyruvate transaminase is converted into alpha ketoglutarate plus L-alanine. And L-glutamate and oxaloacetate in presence of alanine aminotransferase or glutamate pyruvate transaminase gets converted into alpha ketoglutarate and L-aspartate. Decarboxylation. Decarboxylation means removal of carbon dioxide molecules from the substrate. So these reactions are catalyzed by decarboxylases enzyme. This again depends on pyroxyl phosphate. Serotonin, that's 5-hydroxytryptamine 5-HT, produced from the tryptophan, is a neurotransmitter. It regulates sleep, behavior, blood pressure, etc. Histamine is a vasodilator. It lowers the blood pressure. It also stimulates the gastric cell secretion involved in the inflammation and other allergic reactions. So here 5-hydroxytryptamine is acted upon by the decarboxylase enzyme. It causes removal of a carbon dioxide molecule from the 5-hydroxytryptamine and is converted into 5-hydroxytryptamine, that is serotonin. And glutamic acid is acted upon by the decarboxylase enzyme. It will be these to remove a CO2 molecule from the glutamic acid and leads to formation of gamma aminobutyric acid or GABA. GABA is the inhibitor of the spinal synaptic activity. Citric acid is a substrate on which the decarboxylase enzyme acts and it causes removal of CO2 molecule from citric acid and leads to give rise to taurine. So, glutamate on decarboxylation gives GABA. GABA is an inhibitory neurotransmitter. The synthesis of catecholamines, that is dopamine, norepinephrine, and epinephrine from tyrosine, again requires pyroxyl phosphate. Catecholamines are involved in metabolic and nervous regulation. The synthesis of niacin coenzymes, NAD and inhibitors from the tryptophan requires again pyroxyl phosphate. Kynureninase is an enzyme which also requires a pyroxyl phosphate. The tryptophan is converted into 3-hydroxykynurenin. The pyroxyl phosphate kynureninase acts on this hydroxykynurenin and gets converted into 3-hydroxyanthranilic acid. It also gives rise to NAD plus and NAD2 plus. So when this enzyme peroxyl phosphate kynureninase is absent, the 3-hydroxykynurenine is converted into xantharic acid and increased in vitamin B6 deficiency. The synthesis of niacin from tryptophan is impaired. Xantharic acid produced in high quantities is excreted in the urine. It serves as a reliable marker of B6 deficiency and decreased hemoglobin levels associated with hypochromic microcytic anemia is seen in B6 deficiency. So, pyridoxal phosphate is required for synthesis of G-aminolevulinic acid, the precursor of the heme synthesis. The synthesis of niacin coenzyme with NAD plus and NAD plus from the tryptophan is again dependent on pyridoxal phosphate. The enzyme kynureninase requires PLP in B6 deficiency. In the B6 deficiency, The hydroxyanthonic acid is diverted to xanthonic acid, and there will be increased excretion of the xanthanate seen in the urine. Pyroxyl phosphate plays an important metabolic role in metabolism of the sulfur-containing amino acids. Transsulfuration. Transsulfuration is a reaction where the transfer of sulfur group from a homocysteine to serine during the synthesis of cysteine by PLP-dependent cysteine synthase. So taurin again is a decarboxylated by PLP dependent enzyme and decarboxylases, and the product formed here is cysteine, and it is involved in the conjugation of bile acids. Deamination is hydroxyl group containing amino acids. It also requires a paradoxal phosphate acid coenzyme. Pyridoxal phosphate plays an important role in metabolism of sulfur-containing amino acids. That is transsulfuration from homocysteine to serine. This occurs in the synthesis of cysteine, PLP-dependent enzyme, cysteine synthase, and also deamination of a hydroxyl group containing amino acids require pyridoxal phosphate. That is, serine is converted to pyruvate in presence of ammonia. And this is again occupied by the enzyme dehydratase in presence of pyridoxal phosphate. So serine is synthesized from glycine by pyridoxal phosphate dependent enzyme hydroxyl methyl transferase. So the enzyme glycogen phosphorylase contains PLP. Its structure is stabilized by PLP for effective enzymatic function. PLP is again needed for absorption of amino acids from the intestine. Adequate intake of B6 is useful for peroxide of the hyperoxaluria and useful for and also urinary stool formation. So recommended dietary allowances. For an adult, B6 vitamin is recommended in a dose of 2 to 2.2 milligram per day. But during pregnancy and lactation, also range, the intake of 2.5 mg per day is advised. Dietary sources. The animal sources such as egg yolk, fish, milk, meat are rich in vitamin B6. Wheat, corn, cabbage, roots, and tubers are good vegetable sources. So the wheat, corn, cabbage, beans are tubers are good vegetable sources. So deficiency symptoms, um, neurological symptoms such as depression, irritability, nervousness, and mental confusion. All these neurological symptoms are seen in deficiency of vitamin B6. The convulsions and peripheral neuropathy is also observed. There will be decreased synthesis of biogenic amines like serotonin, GABA, norepinephrine, and epinephrine. In children, vitamin B6 deficiency with a drastically reduced GABA production results in development of convulsions and epilepsy. Decrease in hemoglobin levels are associated with microcytic anemia. The synthesis of niacin coenzymes at NAD plus and NAD plus from tryptophan is impaired in case of vitamin B6 deficiency. The high amount of xanthanic acid is also excreted in urine. There is a dietary deficiency of pyroxene is rare because most of the vegetables contains vitamin B6. And it's mainly this deficiency particularly seen in women taking oral contraceptives and in alcohols and in infant born to malnourished mothers. Drug-induced vitamin B6 deficiency, isoniazid combines with pyroxal phosphate to form inactive hydrazone derivatives which inhibit pyroxal phosphate dependent enzymes. Tuberculosis patient on long-term use of isoniazid develop peripheral neuropathy which responds to vitamin B6 therapy. So what happens in tuberculosis patients? Long-term use of isoniazid causes deficiency of vitamin B6, which leads to development of peripheral neuropathy. So in that condition, when vitamin B6 is supplied, then the situation will improve. Even in because isoniazid it combines with the pyroxyl phosphate of the vitamin B6 and make it inactive form hydrazone, which inhibit the PLK dependent enzymes. So the patients with tuberculosis patient who are on anti-tuberculosis drug has to so considering usual treatment of patients with rheumatoid arthritis, Wilson's disease, and vitamin antagonist.